In this video, I'm going to provide a brief overview of the nervous system and its structure and function. A short explanation of the nervous system's function is that it sends and receives messages between your brain and your body using both electrical and chemical signals. These messages allow your body to sense and respond to things as they change inside you and also out in your external environment. It also controls how you learn, what you remember, what you think about, and even your own self-awareness, your ability to sort of step outside yourself and think about yourself. Your nervous system regulates or controls both the voluntary, the things you can consciously change, and the involuntary processes within your body. Basically, there are three parts to how your nervous system functions. You first have the sensory part. This is the information coming in. It's what you are receiving. And there are particular neurons known as sensory or afferent neurons that are responsible for taking in that information. They monitor changes in the environment and then send that information to the brain via the spinal cord. Then there is the integrative or the deciding part of the nervous system function. And there you have neurons that are known as interneurons or association neurons, primarily in your brain, where the sensory information that has come in on sensory neuron is then analyzed and combined together with other information from other neurons so that your brain can create sensations, add to your own memory, produce thoughts or perceptions about what is going on. And then finally, you have the motor or the reacting part of the response, again with another neuron, a motor or an efferent neuron. And these neurons take the nerve impulses from the brain and transfer them to muscles or to glands to create a particular response based on the evaluation that's happened from the sensory information coming in. To put this in a picture, the stimulus here is a glass of water. The sensory neurons in your eyes are able to receive the information that the glass is sitting there on the table. Information is sent, in this case, probably to your brain because you need to decide, do I want to drink that? Do I want to pick it up? The decision is made and the answer is yes. And so then more information goes out through the motor neuron to the muscles of your bicep and your forearm to pick up the glass and to have a drink of water. There are also sometimes when your brain is bypassed on a reflex. I'm sure you're familiar with some of the reflex tests that happen at the doctor's office. And in this case, the information in goes immediately to an action motor neuron without passing through the brain. That's why it's an unconscious reaction that's happening. We'll talk more about the difference between reflexes and responses in a lab that we're going to do later on in this course. Your nervous system has two main divisions. There is the central nervous system, also known as the CNS, which consists of your brain and your spinal cord. And again, that's where most of the thinking, the processing, the integration is happening. And then you have your peripheral nervous system, or the PNS, which are all those nerves that are coming out of the brain and going into the body. Primarily, your nerves have two divisions. They are either coming from the head, the cranial nerves, or off the spinal cord, or the spinal nerves. Your peripheral nervous system can be further divided into the nerves that are sensory nerves that are taking in information, and there are motor nerves that are causing actions in muscles or glands. And then out of the motor division, we have the part that's the somatic nervous system under voluntary control and the autonomic nervous system under involuntary control. We'll talk more about these particular divisions in detail in future lessons. Right now, I just wanted to introduce the idea of how we divide up our discussion of the nervous system and its various parts. So let's talk about the functional unit of the nervous system, the cell that does all the work. The nerve cell is known as a neuron, and this is sort of your basic illustration of a neuron. We have the cell body here in the center. That's where we're going to find the nucleus and the mitochondria, or at least a lot of the mitochondria, and the various other organelles. From the cell body, we have various branching structures. Those are known as dendrites. And the dendrites is where information comes in. So information is coming in on the dendrite end of the neuron. The nerve impulse will flow from the cell body down through the axon, which is kind of like a long tail, to the axon terminal or the end. And this is where information is going to come out of the neuron onto the next neuron or onto a particular muscle or gland. I found this particular analogy to help you picture one neuron. It says if a neuron cell body is the size of a tennis ball, then the axon itself would be a mile long, but only in half an inch wide, and the dendrites would fill a bedroom. Nerves themselves are made up of many, many neurons, but the neurons inside the nerves, primarily what we have running through our body, are these axons, the long, skinny tail between the cell body at one end and the terminal branch where the information is passed on at the other. Many of the axons on our neurons are coated with a particular substance known as myelin. So myelinated axons are important because they transmit a nerve impulse faster, up to 50 times faster. Most of the neurons found in the body in the peripheral nervous system are myelinated so that the information gets around the body quickly. And when we look at the brain, we'll be looking at both white areas and gray areas in the brain, and the white areas in the brain contain these myelinated cells. So the myelin sheath, or sometimes they're known as Schwann cells when we're talking about the neurons in the peripheral nervous system, these are extremely important for making the speed of transmission happen much more quickly. The reason being is it lets the nerve impulse basically jump along from one section of sheathed axon to another instead of having to pass all the way down through every little bit of membrane of that axon. We'll talk a little bit more about that when we talk about the nerve impulse. Neurons come in a couple of different shapes, and I just wanted to point this out. What is most common are these multipolar neurons, or that shape I was showing you on the previous slides. This is what most of your neurons will look like. The cell body has lots of dendrites coming out in various places, and then the long axon to the terminal branch. Many of your sensory neurons are known as bipolar neurons, and in that case you have only one dendrite coming out of the cell body with its branches at the end. And then there's a special type called a pseudo-unipolar, and these sensory neurons have a situation where the cell body sort of seems like it's on the side, and it puts out an axon that in one way it acts as a dendrite, and at the other side that axon is again acting as an axon. So just to give you an idea that neurons come in different shapes. There's also another whole category of cells in your nervous system that are not involved in transmitting electrochemical impulses, the nerve impulse. These are your neuroglial cells or neuroglial cells or glial cells. You'll hear all of those pronunciations. And their job is to support and nourish and protect the neurons. They're involved in keeping the situation around these neurons one of homeostasis, where everything is in balance. Also can help with communication and protection against diseases. There are actually many more of these types of cells than there are of neurons. If we look at our slide illustration here, we can see the big blue motor neuron showing, but these little dots are the nuclei of all of these glial cells. And so in any bit of nervous tissue, you have a lot of these supporting cells, of which there are several different types, and we'll talk about them a little bit more when we look at the individual central and peripheral nervous systems. Neurons have three important characteristics that allow them to do the function of the nervous system. The first characteristic is that a neuron is irritable, or sometimes it's called excitable. It has the ability to respond to a stimulus from the outside environment and convert that stimulus into an impulse or an electrochemical impulse. A second key characteristic of neurons is that it has polarity, that the surface of a neuron, the cell membrane, is negatively charged due to an unequal distribution of ions across the membrane. So there is more positive charge on one side of the membrane, more negative charge on the other. This is primarily caused by a structure known as the sodium-potassium pump that moves those two ions into and out of cells. And then the final characteristic of a neuron is that it's conductivity. It has the ability to pass on this nerve impulse through the electrical chemicals and signals down that particular cell and then send it on to the next neuron in line. Without these three characteristics, your nervous system would not be able to function. I want to just talk briefly about our sensory receptors because, of course, that's the first part of the nervous system response. And we divide our sensory receptors into different categories based on what kind of stimulus will cause them to create a nerve impulse. There are thermoreceptors that react to changes in temperature. There are mechanoreceptors that are activated by mechanical pressure or physical distortion of your body. There are chemoreceptors. These sensory neurons are activated by changes in chemical concentrations, can be both external to the body or internally inside cells. There are photoreceptors, specifically in your eyes, that are activated by electromagnetic radiation, otherwise known as light. And then we have nociceptors that are activated by tissue damage. This is what causes us to perceive something as being painful. We believe that the pain is coming from the cells themselves being damaged. This table gives you some more specific examples of these different types of sensory receptors. So starting with our mechanoreceptors that are responding to mechanical change or physical change. Of course, you can imagine that this would include the touch receptors in the skin, but it also includes the hair cells that are found inside your ears that are involved both in hearing and in balance. Also, receptors that are known as baroreceptors that measure how much your blood vessels stretch to get an indication of your blood pressure. With your muscle spindle receptors, keeping track of how much those muscles themselves are stretching to prevent any kind of tearing. Same thing with some of your tendons. You don't want to stretch too far because you want to preserve those connections. Your thermoreceptors are taken care of by what's known as free nerve endings in skin. So they don't have a special structure, but we just have nerves, as is the nociceptors. They're also basically just the end of nerves in your skin. Photoreceptors, you've probably heard of rod and cone cells in your eyes, in your retina, that respond to light, to photons of light. Chemoreceptors, specific examples there is your sense of smell and sense of taste. And then your blood glucose is also detected by a chemoreceptor because it changes in that concentration. And then this last type of receptor that was not on the previous page, osmoreceptors, are ones that detect how much concentration there is of particular substances in the liquid part of your body, or primarily in your blood. And this is important because you want to stay in homeostasis for hormones or electrolytes and various other chemicals. And so you need a receptor to kind of keep track of those concentration levels. I want to end by talking a little bit about the nerve impulse itself. In short, the nerve impulse is an electrical impulse that moves from the dendrites, or the cell body of a neuron, through the axon to the axonal terminals at the end, to the very, very end point, which is known as a synapse. At the synapse, chemicals known as neurotransmitters are released, and they bind with receptors on the next neuron. We call that the postsynaptic neuron, the next one in line. These neurotransmitters can be excitatory, make the neurons more likely to pass on an impulse, or inhibitory, make the neurons less likely to pass things on. Example of an excitatory response would be when you have caffeine, your nerves are just a little more excitable, they're a little bit quicker to react, which helps you feel more awake. But then an inhibitory response would be something like Botox, which is used to smooth out wrinkles and relax muscles because it slows down the response and the nerves are not able to fire, they're not able to send a message to contract. This is a very complicated chart, but I want to talk through it. Basically, your body is existing, your cells are existing, we're starting down here with 1 on the bottom left-hand corner, in a resting state where the extracellular fluid, the outside of the cell, is more positive than the inside. And we can measure this difference in negative and positive charge by putting an electric voltage on it, or millivoltage. And it's actually at negative 70, so it's not on this chart, but we're starting at negative 70 at the resting potential. So if a neuron is not receiving a message to fire, it's not getting any input from sensory information, the membrane electrical potential is negative 70 millivolts. We can see also illustrated here, we've got various sodium and potassium channels, channel proteins there in the cell membrane. These are closed. You see the gate is closed across the top so that things cannot go in or out. Separate to these channels is a pump over here. So we've got our sodium-potassium pump that is busy moving sodium out and potassium in to keep the millivolt resting potential at -70. When a stimulus comes in from one of your sensory neurons, it is going to change the electrical potential by opening some of the gates. You can see the sodium is now able to move in because the gate has been opened. So it's going to make the outside less positive compared to the inside, and it's going to change then the voltage. It's going to raise the voltage eventually to a -50. -50 is known as the threshold level. This is the level that a nerve impulse will fire because it has depolarized enough of the membrane. It's made the membrane enough more positive that it changes and sends an electrical impulse down the rest of the cell. It can be a slow rise. You can see that the change is very, very slow from the resting potential. But then we have a very, very rapid response afterwards because basically many, many more sodium gates will open and sodium is just going to flood in to the inside of the cell. It's going to really change the negative-positive balance. It's going to depolarize it, make it much more positive. And we see on this little diagram, we see this peak of action. That's when that electrical impulse is going down the cell. We call that the action potential. It becomes so positive, it jumps all the way to +50. So we go from -50 all the way up to +50 in electrical potential, telling us that the cell membrane is becoming much more positive, much less negative, much less difference between the inside and the outside. Then after that electrical impulse has gone all the way down the axon, things reset. The gates are going to let the sodium close the gate so the sodium can no longer travel in. Potassium is let out. That's where we see this curve goes back down. And the membrane basically overshoots a little bit at the bottom, which is why you are not able to have an impulse go backwards. It only goes forwards because after it passes through the axon, that part of that membrane is not able to respond to any changes. And then eventually things will go to reset back. So the main point I want you to gather from this is that it is an electrical change between the amount of positive outside the cell and inside the cell. It's controlled by the gated channels. The channels with little doors on them can be open or shut like gates that will keep sodium, particularly sodium ions, outside of the cell or let it move inside the cell. When those gates are open, that is when the nerve impulse is sent because that's when that electrical charge can be generated by those sodium ions moving into the cell. And then, of course, when we get down to the end of the axon, we've got this electrical impulse coming down the axon, down the membrane. It's going to trigger the release of the neurotransmitter. The neurotransmitter has been made by the cell. It's been stored in vesicles waiting down there at the very, very end of the axon. The synapse is this space in between, so one axon does not touch directly. There's a little bit of space where chemicals are released into that space, and then they float over and are picked up by a receptor on the dendrite of the next axon. So your electrical impulse comes down the axon from the first neuron that is transformed from electrical into a chemical message by the release of these neurotransmitters into the synaptic space, the synaptic cleft between the two neurons. Those neurotransmitters, those chemicals, get picked up by receptors, and then they go on to basically change the electrical potential of the membrane of this new neuron and cause that electrical signal to be sent on, down on through that neuron. Neurons have an all or nothing response. If the neuron is going to generate a nerve impulse, it's going to go all the way. So there's no partial responses. It is, on or off. The nerve impulse will occur whenever this threshold intensity, which remember that was a negative 50 millivolts. So whenever that electrical potential is reached in the membrane of that particular neuron, then a nerve impulse will occur. All of the impulses on any particular neuron are the same strength, but the effect in the body, or the effect on the next neuron, the postsynaptic neuron, the one that's on the other side, will depend on whether there is 1 or 10,000 different impulses coming in from the various neurons connected to it, and if those are releasing neurotransmitters that will cause an inhibitory or an excitatory input. And so it's not the one impulse from one neuron that makes the difference. It's the combination of all the neurons that are firing at any particular moment that determine how your body receives and responds to a nerve impulse. So that covers what I want to talk about in this first video. So to check for understanding, can you describe the function of the nervous system and explain how the three-part process of the nervous response, the sensing, the integration, and the responding, how that is connected to the overall function? Can you identify the major structures of the neuron, the dendrites, the axon, the cell body, and explain how each part plays a role in the nerve impulse transmission? Can you give examples of the different types of sensory receptors and what sort of things they are evaluating and regulating? And can you basically list the steps in the nerve impulse and explain how both electrical and chemical messengers are needed to allow neurons to communicate?